So Clayton, very excited to hear your talk actually on viral proteins. So Clayton Moore completed his master's in bioinformatics at the University of Guelph, where he developed a computational modeling protocol to analyze the structure and function of novel viral proteins. His data-driven mindset led him to Novor, where he's helping researchers better understand and characterize their most important proteins. So Clayton, welcome and take it on.
Hello, everyone, and thank you for stopping by this presentation today. My name is Clayton Moore from Novor. And today we're going to be talking about some applications of proteomics, specifically antibody sequencing and how it can act as a tool to improve reproducibility in the life sciences. Today, I'm going to focus on antibodies specifically because of their sheer ubiquity in the life sciences and just how much we rely on them for our day-to-day research. But it's important to note that the same proteomics approaches we discuss can be applied to really any protein to further characterize it. I want to start out with a simple question. Would you work with a gene, a vector, or really any oligonucleotide with an unknown sequence? Today, I think that the majority of scientists would answer with a resounding no. Biology has become a much more quantitative field and is becoming increasingly digital and more defined. The availability of genomic sequences in public databases allow the same genes to be effectively and reproducibly synthesized and studied in different labs across the world. When a sequence is available, there's really no uncertainty. If I were to order a gene, a vector, RNA, or some other oligonucleotide, I can know that it will be largely indistinguishable from the ones that you describe in your experiments. This allows me to attempt to reproduce your experiments. And while we may have some small differences in the protocols that we use, at least we can be sure that the underlying biological materials are essentially identical. And yet, for one reason or another, we don't hold our research antibodies to the same standard. Even though they've been instrumental in addressing numerous biomedical research questions, they are never defined at the molecular level. We've been using polyclonal antisera for nearly a century and monoclonal antibodies for over 4 decades, and yet the problem remains. Antibodies are characterized by what they bind, rather than their actual physical properties. Now, you may be sitting there thinking that, well, that's the way it's always been, and it's always been fine. But time and time again, we see that this is a serious and ubiquitous problem. Just to briefly illustrate this, over a period of 10 years between 2002 and 2012, Dr. Glenn Begley and his research team at Amgen investigated the findings of a number of top-tier publications. They found that findings from 47 out of the 53 results were not reproducible either by his team or even by the original researchers. Now, we're not talking about some minor findings buried deep in figure 2. These were the major findings, the so-called core message of the paper that they were not able to reproduce. Now, it's not the fault of researchers. They don't want to present irreproducible results. It's not that they're lazy. It's not that it's their fault in any way at all. It's clear and simple. So often, we simply don't have the tools or even have any way of knowing exactly which antibodies we're working with, just because they're only characterized based off of what they bind. Take, for example, these anti-CDK1 antibodies. You can see the 2 antibodies on the left that are currently being sold as binding CDK1 were actually found to also non-specifically bind CEP152, whereas the antibodies on the right show no cross-reactivity whatsoever with CEP152. What's surprising to me is that Abcam is selling antibodies on both the left and on the right of this figure. So how on earth are you supposed to know if the antibodies that you're working with have any cross-reactivity? Your gut reaction may be pretty much the same as everybody else's in the industry, that you should simply check to see what's available in the literature. But the antibodies that are on the left of this figure have been cited in over 200 papers. And some targets out there have over 3,000 antibodies being sold against them. So how on earth are you supposed to find the best antibody in this sea of thousands? It's hard. And quite frankly, there's huge amounts of batch-to-batch variability, even when you think you found a good antibody to work with. So even though you found a good antibody to work with for this experiment, there's no guarantee that when you have to repeat this experiment next year, 5 years from now, 10 years from now, or when establishing a new lab, that you'll ever be able to get access to the original antibody that you found and verified. Now, what I'm talking about here is not a unique example. We could spend this entire time going over examples of antibodies that are leading researchers astray. Take, for example, the figure here. And so what we're looking at here is antibodies that claim to bind ubiquitin in both the heart and the liver. Now, these are being sold across a ton of different vendors, and what we can see is that despite the fact that they're all binding the exact same target, they have completely different staining patterns, which indicates that they're actually binding completely different targets or a series of different targets. I could go further and further and further into this, but I'm just going to showcase one more example. And if you'd like to learn more, the Antibody Society has an incredible webinar series that explains this issue in much more detail than I can get into today. So for my last example, before going into a little bit more on the solution, let's take a quick look at ERCC1. So ERCC1 was used as a biomarker in the treatment of cancer. This antibody, 8F1, was used to detect low levels of ERCC1, which could predict the efficacy of cisplatin-based adjuvant chemotherapy in lung cancer. So for a while, everyone was using 8F1, plenty of papers were published, and honestly, everything was going quite well. And then something changed in the manufacturing of 8F1. Maybe it was a concentration change, maybe something happened in a recloning step, but the end result of this change were clear. Tumors that had previously shown low ERCC1 staining levels were actually now presenting as highly stained. So ERCC1 became, more or less overnight, a completely useless biomarker. And if that wasn't bad enough, it was actually found that 8F1 didn't even specifically identify ERCC1, but it also recognized CCT alpha, And in the end, it actually turned out that it was CCT alpha that was indicating greater survival outcomes and not ERCC1 at all. And that wasn't discovered for years, and yet researchers were constantly going back to the literature to refer to this specific monoclonal to further research everything. But something changed in the manufacturing process that overturned years upon years of actual hard research. And I know that these might just sound like horror stories, but unfortunately, they're not new and they're not unique. The issues that I've been describing here today have been coined the reproducibility crisis, and we've known about it for years now. It's gotten so bad that we're at a point in which 75% of antibodies don't bind specifically to their target. 56% of published antibodies can't be reliably traced in any meaningful way, and over 50% of antibodies can't even be used in their intended applications. So how on earth are you going to know what to do? Now, all of these challenges result in billions of dollars wasted and countless hours spent on these antibodies that don't even do what they claim to do. So just to review a little bit here before moving on, what exactly is the problem? Well, simply put, it's not any individual thing. It's a complex mixture of issues that all compound on one another. We're using so many polyclonal antibodies, which have been historically and practically undefinable. And beyond that, many monoclonals aren't actually monoclonal at all. We're going to get to that in just a minute here. But there's a large issue in terms of the reproducibility in monoclonals, that they are actually very similar to polyclonals. And hypothetically, let's say that you found a good monoclonal that you like working with. It binds reliably with no off-target binding. Unfortunately, because of this huge batch-to-batch variability problem, just like we saw in ERCC1, something might change in the manufacturing process that you have absolutely no control over, and you would have absolutely no way of knowing this. Ultimately, the QC on these antibodies tends to only be done once during the early stages of the development of an antibody and is never done again, despite the fact there might be some major changes to the actual antibody itself. Fundamentally, this allows for antibodies to be sold with incomplete validation, solely because antibodies are being sold based off of what they bind and not their actual physical characteristics. So, what can we do? Well, for starters, we need to get higher standards when it comes to our key research antibodies. We should value vendors and researchers that are building towards recombinant antibodies and genuinely care about reproducibility. We need to bring antibodies into the 21st century. We need to not trust antibodies that are not recombinantly expressed at the bare minimum, and at the best case scenario, with presented sequence data that defines exactly what the antibody is, rather than simply relying that it binds what the tube says it binds. So, where do we get started? I think the obvious starting point here is going to be hybridoma sequencing. It's become a readily available tool as long as you have access to the cell line, and it's reasonably straightforward. It does have two main drawbacks. Firstly, you do need access to the cell line, and secondly, not all antibodies are actually monoclonal. Researchers, including our own team, have routinely found that many so-called mAbs actually have additional chains. So take this paper published recently by Bradbury et al. They found that over 30% of monoclonal antibodies actually have additional chains and are therefore not monoclonal at all. So if we're talking about a single additional chain, take for example the light chain at the top of this figure, then that will create two unwanted pairing variants. So what we've got here, best case scenario, we might get some lower activity from the antibody that's a mixture, or no binding activity from the completely different specificity. But in the worst case scenario, we can start binding different targets altogether. And these are going to be completely random based off the pairings. And 3 variants may not sound so bad, but that's when we're talking about a single different chain. When we're talking about an additional heavy and an additional light chain, then we only have 1 correct IgG pairing and 9 unwanted contaminants. And it's completely unclear what these unwanted pairing variants are actually going to bind to, and could create some really, really big challenges in the actual characterization of an antibody. Now, these are not rare circumstances. A single additional chain happens in about 30% of cases, and we see multiple chains in additional 3% of cases. So roughly 1/3 of the time, these variable numbers of chains pop up. And these can happen all over the place. When we're working with hybridomas, there's an intrinsic instability due to the fact that we're working with a cancer fusion cell line. The additional chains could come up for many possible reasons. There could have been some chromosome loss or duplication during the hybridoma generation. There could be additional rearrangement of alleles. It could be that the mixture was actually never was non-clonal from the beginning, or it could have had fusion from multiple B cells. Honestly, it's anyone's best guess. And detecting these additional chains in hybridoma sequencing is incredibly challenging, if not completely impossible. Now, what if we actually work with the proteins directly? After all, it's the proteins that are doing the actual work. So it seems like a reasonable assertion. If we can work with the proteins directly, then it sidesteps a number of the issues and even provides some additional beneficial information. First of all, there's little to no ambiguity in terms of the chain pairing. As long as the mixture has been purified, we can confirm the activity, the binding activity that is, in vivo, and we can observe the exact pairing combinations that are present in the tube itself. So this allows us to identify exactly which pairs bind with which. So even when we do have additional chains, like the 30% of cases that we're seeing earlier, then we can handle that with no real problem. We can also look for and identify post-translational modifications that simply can't be observed in hybridoma sequencing because, they haven't been introduced yet into the DNA. So these are things that can only be observed when you're looking at the proteins directly. And some of these PTMs, like O-linked glycans, can have significant impact of the antibody activity, particularly if you observe them in the variable region or the CDRs. And lastly, one of the biggest benefits of this technique of working with the proteins directly is that you don't need access to the actual living cells. There are tons of cases in which either the hybridoma dies, it mutates, or a researcher has been working on this hybridoma library for their entire lives. And once they retire, all of the existing cells are actually thrown out, and all we have left is the protein. And with hybridoma sequencing, there's absolutely no way that we can go about recovering that. But when we look at the proteins directly, even the tiniest amount of protein sample left buried somewhere deep in your freezer means that we could actually have potential of recovering the antibody sequence from a completely de novo approach, rather than having to start the entire library 100% from scratch. And that's exactly what we're trying to do at Rapid Novor. In a word, we sequence antibodies and proteins using mass spec. So that means that we're working directly with the proteins themselves, and we don't need any access to the cell lines themselves. We need no prior knowledge of the antibodies, and we are able to produce reliably high-quality de novo sequencing results from amino acids directly. Historically, this mass spec-based de novo sequencing has been time-consuming, it's been costly, and to be honest, it's just difficult. So what we've made it our mission to make proteomics more accessible to the research community at large. We've dedicated our entire efforts towards offering a de novo antibody sequencing service that is reliable and robust. And in doing that, we've actually brought down the cost about 5-fold over the last 10 years. It's not to say that it's now an easy process. It still does require a large amount of expertise and a deft hand at that. But what we've done is we built off of our platform that was originally developed by our founder and CSO, Dr. Bin Ma, to sequence any antibody in a matter of hours instead of months. In over 2,500 projects, we've seen a lot of unusual samples, to say the least. And using that knowledge, specialization, and experience, we can bring 100% accurate protein sequencing results to every single one of our samples. So, how do we do what we do? Well, honestly, we apply a fairly standard bottom-up proteomics approach. Starting out with a purified antibody, we run a panel of 5 to 10 different protease digestions. We follow that with LC-MS/MS, from which we can sequence the peptides and assemble a full sequence. On the face of it, there's nothing too remarkable about it. But when we look a little bit deeper, then that's when it starts getting really cool. We've spent years optimizing our digests so that we can get some short peptides and some very, very long peptides to ensure incredibly deep coverage at every single amino acid position. We guarantee a minimum of 5 times coverage, but regularly see 30 times to even 100 times coverage at every single amino acid position. And what's really interesting here is that we've developed our peptide sequencing algorithms so much that we can actually be sequencing everything pretty much on the fly. That's to say that when a sample is on the mass spec, we are in real time sequencing the individual peptides, which allows us to really optimize for different regions of the amino acid, of the actual full antibody, so that we can minimize the number of experiments that we need to do downstream, because we can adjust the experiment while it's still happening. Combining that with our state-of-the-art assembly algorithms, ultimately we are able to get these incredibly reliable results in, quite frankly, incredibly quick time frames. And using our approach, we've tried to make everything completely de novo. So that means it's actually fairly easy to get a project started. We are completely ambivalent to isotype and species, and we strive for the same level of confidence for every single antibody we see. It doesn't matter if it's mouse, human, goat, llama, shark, really anything. All we need is about 100 micrograms of reasonably pure antibody sample. This holds true for monoclonal antibodies and even the oligoclonal antibodies that we were discussing earlier. There are several additional chains, and when we see multiple heavy and light chains, rather than just abandoning the project and saying that it's too complex to sequence, using our algorithms, we are able to reliably sequence both heavy and both light chains with the same level of confidence. What we can do this with is really up to 5 antibodies in a mixture, so long as they're similar in molar ratios. So that's to say, if we can get about 100 micrograms of 5 different antibodies in a mixture, then we can confidently sequence that with 100% accuracy. But if one of the antibodies were to outnumber the other antibody, let's say 100 to 1, then we would really just be sequencing that dominant form here, because the remaining antibodies are likely just background information. So fundamentally, what this means is that the challenges of multiple chains and samples has largely become a non-issue for sequencing using our platform. And I don't want to give the impression that any of this analysis is simple or mundane by any means. We've spent the past 5 years building extremely strong algorithms and a highly specialized team to get all of this work done in a very, very short period of time. So our standard turnaround time is just 2 weeks, but in a pinch, let us know and we can try and work around your schedule. For example, I think our record right now is about 26 hours from when the sample originally got into our lab to a full report generated and delivered to a client. Now, it's not to say that we can promise that for absolutely everything. But for some high priority projects, give us a call, see what we can do. But what you should typically expect is about 1 to 2 weeks for a single sample. But our workflow actually scales really, really strongly. So we can get up to 10 samples done in closer to 2 to 3 weeks, 20 in 3 to 4 weeks, and so on and so on. And if you have a particularly large batch, just let us know, and then we can see what we can do for you. And that being said, we will never sacrifice sequence quality for a quicker turnaround time. When you get the sequence of an antibody wrong, it is immediately apparent during binding. So we always do our absolute best to ensure that we are getting 100% accurate results on every single project. And we do that through a number of ways. And we can keep doing this with our robust turnaround time because all of our facilities, that is all of our lab techs, all of our software engineers, all of our bioinformaticians, all usually work under the same roof. Obviously, right now, it's a little bit more challenging and we are more spread out. But we still have a highly collaborative environment in which our lab techs work on site with our 8 mass specs so that we can really order additional experiments incredibly quickly to ensure that we have a guaranteed result on every single one of our sequences. We do that in a number of different ways. So we guarantee a minimum of 30 times coverage on every single amino acid position. That's in the CDRs, in the constant region, absolutely everywhere. We want to make sure that every single amino acid is verified with multiple different coverage reads from multiple different protease digestions. And we are going to review that over and over again. So even though our initial stage is built off of this AI-driven large data approach, we make sure that individual bioinformaticians manually review every single sequence so that we have no ambiguity in terms of the actual sequence. And lastly, we make sure that our bioinformaticians are looking for any isobaric amino acid combinations. So take, for example, here, the gel on the left. This is not something that we want to see all of the time. It's a bit of a bizarre situation, but I think it illustrates exactly the work that we can do. So it came from a postdoc that was trying to wrap up some of her work before leaving the lab, and she was quickly running out of antibody. The cell line was long gone, and she was down to the last few micrograms of the actual antibody. And that's when she reached out to us. We started the project and we ran it on a gel. But I'm going to be honest, it doesn't look great. Even when you squint, you can't see much of an antibody in there. But nevertheless, we gave it a shot. We purified it, we ran our standard protocol, and in just 2 weeks, we delivered a sequence with strong coverage on every single amino acid with every single residue confirmed. I'm not saying here that we want all of these samples to look like they do on the left here, but even with a sample that was that messy and that contaminated, we were able to get coverage results that were unthinkable even just 10 years ago. And Although our process is going to start out with an AI-driven deep learning algorithm, we want to ensure that every single sequence is independently reviewed by a minimum of 2 bioinformaticians. They review the spectra to confirm every single residue and do isobaric checks looking for combinations of amino acids that even weigh the same on the mass spec. One of the most common challenges here is actually isoleucine and leucine. At a first glance, they have an identical molecular mass, 113 daltons. So they're indistinguishable on the mass spec, right? But despite this, they do have unique R groups. So what we can do is fragment these individual amino acids further, where they will lose a very specific amount of mass in form of a W ion. We can measure this loss in mass, and from this data, we can determine whether the residue is an isoleucine or leucine with unmatched certainty. We call this WILD, for W ion isoleucine leucine determination. Traditional approaches here are going to be more frequently looking for germline sequences from which they can look at residues. They are going to rely on different digestion rules, but neither of these approaches are truly de novo. And when we're regularly seeing 5 isoleucines or leucines in the CDRs, the accuracy becomes incredibly important. We want to have every single residue confirmed based off of data rather than our best guess. So at Rapid Novor, our goal is to deliver a single sequence that just works that first time. Leave the complex analysis to us, and we want to make your life as easy as possible here. Now, I could go on and on and on here, but I think one of our clients really said what's taking me 30 minutes to say in one sentence. They offer best-in-class service for de novo sequencing with low material requirements and extremely fast turnaround times. I mean, I might sound biased, but give it a shot, and I promise you that we can hold up to this legacy here. I don't have much more time, so I'm just briefly going to mention one of the latest developments here at Rapid Novor. I've mentioned a few times already that polyclonal antibodies have largely been undefinable, but by applying a proteogenomic approach to sequencing, we've actually been able to completely de novo sequence 2 binding monoclonal antibodies from a parabbit polyclonal mixture, plus 1 monoclonal antibody that was selected at random from the pool, and a 4th one that acted as a negative control. And we found that when we expressed these sequences that we derived from the polyclonal workflow, that the actual recombinant antibodies largely have better binding activity than the original polyclonal mixture. At this point, we've developed a repeatable process that can reliably sequence polyclonal mixtures by supplementing a similar proteomics workflow to our monoclonal sequencing platform in tandem with NGS data. And researchers are already using the sequencing platform for a variety of purposes, including therapeutic antibody development, further characterizing complex immune responses, biomarker discovery for diagnostic development, and, like we saw in the previous slide, converting a polyclonal mixture to a single monoclonal. And although the platform is already showing reliable and reproducible results, we hope to further expand its capabilities in the months and years to come. These developments include custom NGS developments so that we don't need to rely on model organisms, and eventually a full de novo sequencing platform that relies on protein-only protocols to fully characterize existing polyclonal samples. But for the time being, we're focusing on our efforts on generating more hits with broader epitopic coverage. So if you have any questions about polyclonal sequencing, about monoclonal sequencing, or really any applications here today, feel free to send me an email, give me a call or text, or you can reach out to our website, rapidnovor.com, with any additional questions. Thank you so much, and have a great day.
Wonderful. That was a great presentation, Clayton. And if you are in the room, there's 2 questions for you. The first is from Dr. Appingbay, Senior Scientist from APTRO. Dr. Bay is asking, can your platform work on any protein sequencing beyond antibody?
Yeah, that's a really good question. So I focused here on antibodies because it's our core specialty. But yeah, we can apply this similar proteomics approach that can work with really any protein. It's just going to depend on the size of the protein and based off of how we target here. So we specialize on antibodies because of their nice predictable structure with the constant domain and variable region. But we can apply the same proteomics techniques to an entire protein. It's just with larger proteins, we occasionally need a little bit more sample just to make the molar ratios work out so that we can get reliable coverage data at every single amino acid position so that we can offer the same high quality standards with infinitely variable proteins.
Wonderful, wonderful. And there's one more question for you, Clayton. It's from Dr. Steve Miller, CEO of Sandia Biotech. He's asking, well, he's saying brilliant presentation showing challenges of old antibody tech for diagnostics. Only need to sequence variable, heavy, and light chain regions. So how fast?
So one of the advantages to our platform here is that we're capturing data incredibly quickly. So it's the limiting factor tends to be mass spec time rather than the actual sequencing assembly. So I was talking earlier about the different sizes of proteins in comparison to antibodies. So all of that happens at the same kind of general time frame. So it doesn't take us any longer to sequence just a fragment of an antibody compared to an entirely large protein. It really just all comes down to the data analysis. So we'd be looking at, like I said, our record time is about 26 hours. So in terms of our general throughput, we would be looking at any protein of any kind of reasonable size within 1 to 2 weeks and combining up to 10 in just 3 weeks.
Wonderful, wonderful. And if there's any other questions for Clayton, and if you're interested in connecting with him and his team from Rapid Novor, you can do so by using our meeting scheduler link. You can also email myself and we can connect you with them as well. And Dr. Dust, do you have any comments or remarks for Clayton?
No, I don't. Thanks a lot, Clayton.
Thank you.
Perfect.
And thank you again, Clayton. And we want to thank our sponsors again for participating. And Steve Miller did say, please contact him ASAP.
Will do.
Awesome. Awesome. So we'll be all heading over to the next session by Dr. Molson, professor. He'll be speaking next.